Chapter 11, Biotechnology, Principles and Processes. In this chapter, I've already made 3 videos. This is video number 4. And if you all have not seen the previous video, do see that video first. In this video, I'll be talking about vectors for cloning plants and animals. In the previous video, already talked about ORI, selectable marker, and cloning site. Now, children, you know that since ages, we know that when bacteria or viruses attack plants and animals, they transfer their genes into our cells and force our cells to produce what these bacteria or viruses want. So, in biotechnology, there are genetic tools of some pathogens is used to deliver gene of interest. That is Agrobacterium tumefaciens, which is generally used in dicot plants and retroviruses. Agrobacterium tumefaciens, it is a pathogen of several dicot plants and is able to deliver a piece of DNA known as tDNA, and then it transforms the normal plant cell into a tumor to produce the chemical required by the pathogen. Now, what do we do? You can see over here the tDNA present in the plasmid of Agrobacterium tumefaciens. This plasmid is known as TI plasmid, that is tumor-inducing plasmid. Now, we remove this tDNA. This tumor-inducing TI plasmid of Agrobacterium tumefaciens is modified into a cloning vector. How? By removing this tDNA. So now it is no more pathogenic to the plant, but it is still able to use the mechanism to deliver the gene of interest into variety of plants. Now you can see over here, instead of tDNA, we put our gene of interest. Now the second is the retrovirus, the second vector. Retroviruses are what? These are the viruses which have RNA genome, and these retrovirus in animals have the ability to transform the normal cells into the cancerous cell. Now this retrovirus is disarmed. The genes producing the cancer, it is removed, and in place of that, our gene of interest is put, and it is used, this retrovirus is used to deliver the desirable gene into the animal cell. Once a gene or a DNA fragment have been ligated into a suitable vector, it is transferred into bacterial, plant, or animal host, where it multiplies. Now, the next thing, let us understand what is gel electrophoresis. It is a technique of separating the DNA fragments based on their sizes. Children, in the previous videos, I've told you that we take restriction in enzymes, which cuts the DNA at specific site. And now we have to separate and isolate this DNA fragment. And this separation and isolation is done with the help of gel electrophoresis. Now we all know that DNA fragments are negatively charged, and it can be separated by forcing them to move towards anode under an electric field through a matrix. In the gel electrophoresis, the commonly used matrix is agarose gel, which is a natural linear polymer extracted from a seaweed. The DNA fragments are separated according to their size because this agarose gel has a sieving property. So what happens? The smaller DNA fragment moves ahead, and the bigger one, they are behind. So you can see the wells also over here. The sample is put into these wells, and then it is connected to an electric field where the smallest fragment moves farthest. Now, the separated DNA fragment, how to see it? It is visualized after staining the DNA with ethidium bromide, and then it is followed by UV radiation, because you cannot see the DNA. So under UV radiation, you can see as bright orange band. Yeah, you can see that. the bright orange bands. And these are the smallest fragment because it has moved farthest. The separated bands of DNA are then now last cut out. You cut it from the agarose gel, and that is known as elution, cutting of the DNA. The purified DNA fragment are used to form a recombinant DNA, and it is joined to the vector DNA. Now you get what? A recombinant DNA. So this is how you separate and isolate the DNA fragment and then add it to the vector, which is also cut by the same restriction endonuclease. Now let's talk about competent host. This is the third and the last two. Now this recombinant plasmid has to enter the host. The host can be a bacteria, it can be an animal cell, or it can be a plant cell. Now, we all know that DNA, being hydrophilic molecule, cannot pass through the cell membrane, as cell membrane is hydrophobic. So how will the bacterial, plant, or animal cell take up this plasmid? They will take it up in different ways, like talking about bacteria. In this, what happens? In order to force the bacteria to take up the plasmid, first thing, the bacterial cell has to be made competent to take up the DNA. Now, how to make the bacterial cell competent? The bacterial cell is treated with specific concentration of divalent cation, such as calcium ion. Okay. So we treat it with calcium chloride. You can see over here, treating it with calcium chloride. Now, doing so, what happens? It increases the pore size of the cell wall. The bacterial cell with the recombinant DNA now is placed on 42 degrees Celsius. First is placed on ice and then place it on 42 degrees Celsius, giving it a heat shock. So first you place it on ice, then at 42 degrees Celsius, then again on ice. So that is a heat shock treatment. And this enables the bacteria to take up the recombinant DNA. Next, other method is microinjection. It is used to introduce the recombinant DNA in animal cell. In this method, the rDNA is directly injected into the nucleus of the cell. You can see over here, microinjection, where it has been injected. Then next is ballistic or gene gun. This method is useful for plant cell. The cells are bombarded with high velocity. Microparticles of gold or tungsten is coated on the rDNA. Now children, when microparticles of gold or tungsten is coated on rDNA, then the rDNA mass increases and you give a very high velocity. So the momentum increases. And then it goes directly into the nucleus. And the third is the disarmed pathogen. Now, in this disarmed pathogen vector, when they infect the cells, they transfer the rDNA into the host like viruses and agrobacterium tumefacium. This I've just now talked to you, explained you about. Now, with this, we finish up all the tools. Now, the next topic which we have to study is what is the process of recombinant DNA technology? There are 6 processes: isolation of genetic material, cutting of DNA at specific location, amplification of gene of interest, insertion of recombinant DNA into the host cell, obtaining foreign gene product, and downstream. In this video, I'll be explaining the first 3. The first one is isolation of genetic material. We know that we have to take out the DNA and cut it with the restriction enzyme. But before that, you have to remember that the DNA is within the nucleus and the nucleus is in the cell. So you have to dissolve the cell membrane, the nuclear membrane, and then take out the DNA, and the DNA in a purified form. And that DNA also has histone proteins, In the nucleus, there are RNAs, so you have to dissolve all those things. So to cut the DNA with restriction enzyme, purified DNA is required. Now, in a bacterial cell is treated with enzyme lysozyme, plant cell is treated with enzyme cellulase, fungal cell is treated with chitinase because the cell wall is made up of chitin. RNA is removed by treating it with enzyme ribonuclease, and the proteins are removed by treating it with protease. After several treatments, you get the purified DNA, and that is precipitated by adding chilled ethanol. And then the fine threads of the DNA, which you can see over here, you get these fine threads of DNA. Now the next is cutting of the DNA at specific location. The cutting of DNA, obviously, that we have already done in video 2, that we use restriction enzymes. The purified DNA is incubated with specific restriction enzyme at conditions optimum for enzyme to act of this. And then the process is repeated with the vector DNA also. Then after that, isolation of the DNA, which I have just now explained you, with the help of gel electrophoresis is done. The activity of the restriction enzyme can be checked. Since the DNA, I have told you, is negatively charged, so the smallest part, the cutout part, moves towards the anode, and then elution is done. So I have done 2 processes in this video, that is isolation of genetic material and cutting of DNA at specific location. The rest 4 is coming up in the next video. Thank you. And if you all have not seen the previous video, do see it.